of the nervous system. The nervous system really depends on the organisms. It's really interesting if we look at animals throughout the animal kingdom, how some animals have brains and some have a brain-like system, and some of them have more than one brain-like system, and some of them have no brains. And we all of us animals function so well on survival that we know, animals, we think when animals are approached by some kind of negative stimuli, they're very reactionary, and that's true whether you have a complete brain or not. So, I always think this whole idea of humans being such higher organisms, it's like simple organisms do a lot of the same things we do without even having the advancements that we do. So it's kind of interesting. In humans, we have our brain, we have our neurons, which are the nerve cells, and then the spinal cord in the middle. So we're going to focus mostly on the neuron. The neuron is called, the nerve cell is called a neuron. When you look at stereotypical biology textbook cells, they show a round cell, and the neuron looks nothing like that. It is a really wild structure. We'll just take a look at that in just a minute. The neuron performs 4 main functions to receive information from whatever cell, organ, gland, tissue is contacting it. It integrates that information and then determines whether an output signal will be made. It sends that signal on, if a signal is made, it sends it on to the end of the neuron, and then that is going to transmit a signal on to the next cell. Could be another nerve, it could be a gland, it could be a tissue, it could be an organ, it could be a cell. So I know a lot of times, like in the book, it'll say glands or muscles, but really it could be anything in the body. So this is what a neuron looks like. We've actually got here in this picture, and you have it on the next page, I believe, just if you want to take a look at that. Yeah, 2 pages more. In this picture, what you have is you have remnants of at least 4 neurons and then 1 full neuron. This light orange one in the middle is a full neuron. You can see in this illustration the interaction between this full neuron, right? Looks like, I don't know, almost looks like a space creature. And then up here you've got a blue end of a neuron and a dark orange end of a neuron. And then down here you've got a dark orange beginning of another neuron and a blue beginning of another neuron. So just to point them out. All right, we'll get to the parts now. So flip back and then we'll, you'll write about the different parts. So the dendrites. The dendrites are these branches at the tips that kind of look like chorionic villi. So go back to page 247 for your notes. The dendrites are the tips. They have all these little branches, as I said, look like kind of like chorionic villi. They're receiving information from other neurons, cells, glands, tissues, organs. Something in the environment outside of it, it's receiving information. So the dendrites are just to take in information. When the dendrites take in information, going to send that information into this main part called the cell body. Oh, sorry, I haven't gotten there yet. Here's the thing about the way, if we're looking at the cellular level, and we've talked about we've talked about hormones being ways to communicate what needs to happen next, as we talked about feedback systems, that this hormone is going to contact the next cell which is going to integrate that information and make that cell do something else and produce something else, which will be sent on to the next cell. Hormones are large compounds. When we're looking at neurons, they are mainly dealing with really, really, really small molecules, actually going down further, not even just molecules, but atoms, or especially ions. And so if you remember a little bit about chemistry and ions, an ion is a charged atom or molecule. The hydrogen ion, the chloride ion, the potassium ion. These are very small in comparison to hormones. This is mainly what neurons are dealing with. Neurons, so these pluses and minuses, if you remember back to talking about the EKGs and what you did in the blood pressure lab, that when you had those electrodes hooked up to you or one of your classmates, they are detecting electricity within your body. And electricity is produced by the balance and movement of ions, the positive and negative charges in your body. Sharks, skates, rays, they all have the ability to feel this electricity from these ions. We use a machine to detect those. So that's the way the neurons work, is that these are the signals that are coming into the cell body, and they will determine the amount of, and we'll talk about positive and negatives in just a bit, the amount will tell you whether a signal is sent on. So the cell body is taking those ions and it's doing math, it's adding up how many positive ions, how many negative ions. And based on that ion information and adding up the ions, that's the processing of it. It's just like the neurons doing a math problem. That the cell body is the mathematician of this all. The signal that is pushed on is called an action potential. I like this term, right? The potential to do action. That's the kind of signal that's being sent on. The potential to do action could be sending on a signal that says, tell the next neuron to tell the next muscle to do something. Our neurons, the whole point of our neurons is to respond to stimulus in our environment and to help us to survive when things change. If an action potential is sent on, it goes down this long, skinny part called the axon. Now this is just like a naked neuron here, but neurons typically have insulation. The axons typically have insulation around them so that ions aren't just being lost out of this long skinny part of the neuron. But they want to keep those ions moving so that they can send on a signal to the next, whatever it is, cell. In this case, we're looking at neurons. I think it's interesting. Some of our neurons can be so long, they can be a meter long. So thinking about like our spinal cord, which runs from the foramen magnum up inside our head all the way down, right, to your buttocks, that you've got almost a meter long neuron, large neuron, or neurons that are cabled together in there that are going to send information from your brain down to your lower limbs, right? So if there's a fire and we get a signal that says there's a fire in the north side of the C building, our brains are going to say, okay, we got a little time, it's down there, we can pack up, and our brain is sending information to our legs like, get up and get out. And so we don't even have to program that. We don't have to say, hey, brain, tell legs to do this. Our brain just integrates all that information and does it without us commanding those details. So there's a little, maybe, nicer picture of the insulation. But the myelin, we've heard of myelin sheath or myelin. This is the insulation that covers the axon. One, it helps those ions to not escape or diffuse out of the axon, and then also by doing so, having this insulation, it helps the ions to be focused in a direction where they move faster. So kind of interesting about this, that at the age of 50, your myelin starts to break down faster. There are diseases that are guided by what's called demyelination, the loss of the myelin sheaths. And so, like for example, Huntington disease, Alzheimer's, possibly. I mean, Alzheimer's, if you are really into puzzles and doing research, oh, there is so much going on in Alzheimer's, because we already know there's likely many reasons for Alzheimer's. It's kind of like thinking about different kinds of cancers, that it's probably like there's an Alzheimer's umbrella, that there's a lot of different kinds and ways or mechanisms of making Alzheimer's happen. So a lot of research, there's a lot of exciting things happening right now, especially. Multiple sclerosis is another one, which is on the rise, and we're not exactly sure why or how. So lots of really important research going on, that one of the main reasons for these big diseases is the loss of myelin, and then the loss of the signal from the cell body to the end of the neuron. That while the brain might be saying, I have a friend, she's having some neurological issues, she's not sure why, she's been to a lot of doctors, and she's having problems where, like she said the other day, she went to get her cat, it had run out, she went to get it, and she went to run and her one leg wouldn't move. And she fell on her face. And she goes, I don't know, I would just normally, I would just normally run, and I couldn't. The one leg wouldn't, it just wouldn't move. So something, who knows what's going on, but lots of interesting things going on in the world of neurology, always. When that signal gets to the end, those are called the synaptic terminals. They are swellings at the end, and there is a junction area between two cells where the synaptic terminals meet, called the synapse. So these ends here are the synaptic terminals. You can see they're kind of bulbous at the very tips of the branches. If you take a look at this area between this new neuron and our original neuron, there's actually a space, and look at that, there's receptors. So I know we've been talking a bit about receptors and how you can see in here that the chemical sent on is circular and the receptor end shape is circular, so that when this binds to that, it's going to stimulate this cell to calculate all of its information in the cell body, and then this next neuron cell will decide, do I send on an action potential down my axon or not? So these are the signal transmitters, the junction between two cells that say, hey, you're gonna do something. All right, so let's talk a little bit about how an action potential is produced in the cell body. As we said, the neurons are going to send information and they're going to receive information. And when we're talking about this information, we're talking about those ions or the production of the ions adding up to become some kind of electrical signal. All right, if the input from the dendrites is high enough, and we'll get to that in a second, then the cell body produces an action potential and sends it down the axon. So it's kind of interesting with neurons, it's not a huge necessary range of when an action potential is sent on. It's just kind of a little range to make a difference in sending on messages. When the action potential reaches the synapse, it's going to release a chemical we often call a neurotransmitter. I like that name again, right? Indicates it's a transmitted signal from a neuron, and then it's going to send it to maybe another dendrite, could be a muscle, a gland, a cell, a tissue, an organ. All right, so a little bit about action potentials and how they work. Every neuron is different. Inside of the cell body is typically negative. It has a bunch of negative ions inside, and which, well, excuse me, it has ions inside. Typically, there are more negative ions than positive ions. Okay, so if this is a cell body, so we're taking a look at our example here, that you can see there's a few positive ions, but there are more negative ions. So let's say that in this particular neuron, that our, what we call the threshold, is at -10. And now a signal is received from some other neuron. And let's say it's receiving some hydrogen ions. So if we started here at -60, let's say when everything was added up, that the inside of the cell body was at -60, and that when these signals came in, these hydrogen ions, that let's say the signal got to -22. The threshold for this particular neuron is here at -10. So the cell body is adding up this signal, and it's like, well, we did move in a positive direction, and this is where it gets maybe a little confusing, that you have to realize that going from -60 to -22 is moving in a positive direction, right? We're moving toward positive numbers. While it moved in a positive direction, it did not hit this particular neuron's threshold. So while the signal was received, the cell body was like, nope, I'm not going to send on an action potential because this information that came in was not positive enough. So I don't have to send on a signal to the next neuron, gland, cell, organ, tissue to do anything. It's not a big deal. So let's say that we suddenly smell like chocolate chip cookies. So you're sitting here and you're like, oh, it smells good. Is it going to cause you to get up, grab your stuff, run screaming from the room? We're gonna die, right? If you smell chocolate chip cookies, it's kind of like this. Like, there's a stimulus, but nothing happens. It doesn't cause you to react in some kind of survival stimulus. So now let's say we smell a very, very, very strong, smoky chemical smell. So new signals received, and we get this mass flushing. Of all of these different ions that are positive. And when the cell body adds this all up, we get to -5, which means that we have crossed the threshold. And by crossing the threshold, it tells that the cell body tells the axon, send on an action potential. You are going to tell the next cell to do something. So this is a very, very simplistic way of explaining the way neurons work. That you just, for every particular cell, they have a different threshold. And if, when the information is received from some other place outside of this particular neuron, if it becomes positive enough and crosses the threshold, then a signal is sent on. If the signal is not positive enough to cross the threshold, nothing happens. So only, regardless of whether the threshold is still negative, or maybe it's positive, as long as the threshold is crossed by the signals that are received when they are added up in the cell body, then an action potential is triggered to be sent on. This concept is called all or none. Neurons do not send on strong action potentials versus weak action potentials. A neuron can pass on a particular specific, one neuron can pass on one kind of action potential. It does not have the ability to send on a really, really strong one versus a really, really weak one. So let's say some other signals come in, whatever this green one is, and the green one, let's say it becomes, let's say whatever this green stimulus is, the cell body adds up to positive 30, which means it's crossed the threshold. It's crossed the threshold a lot more than whatever the red one was, but guess what? The same strength of action potential is sent on. Whether we're talking about it just barely passes threshold or really passes threshold, the action potential strength will be exactly the same. And so all you have to do is hit -10 for this particular neuron. You hit -10 or more, action potential is sent on of the same strength. So again, this is called all or none. This is called all or none. Make sure you know that. This shows us that sometimes, and this gets into, we're not gonna get into the details of depolarization and repolarization. It's basically the different ions that will exist in and around the neuron, the cell body. But regardless, when you're talking about just hitting, when the threshold hits, like here and here, you could see that these two, whatever these stimulus were, they're very low in terms of adding up what's happening here, which means they were somewhere here when the cell body added up, so nothing happened. But this one here hit the threshold, right, went up past the threshold, and what happens is that they always come eventually back to, you have the flushing in of ions and the flushing out of ions. So when this all happens eventually, like the red one, after that action potential is sent on, it goes back to its resting state. So then it goes back to normal, and then it's waiting for the next signal. So all of this happens, and then it comes back to where it was to begin with, before that signal was sent. Okay, so how do we get the intensity of a signal to change? Because clearly we react differently to different situations, right? Just like with smell, if you smell something, anytime you smell something, you're not reacting with screaming and yelling and going, we're all gonna die. So intensity of a signal is determined in a couple different ways, and it's definitely not stronger or weaker action potentials. So what has to happen is there's a couple of things that can happen depending on the kind of neuron and depending on the situation. We'll take a look at two different options that can happen to pass on an intense signal, not an intense action potential, but to make us react to a stimulus strongly. One is this signal keeps coming and coming and coming and coming and coming. So let's say that smoky chemical smell that's coming, at first we smell it a little bit, and then we smell it a little more. And what's happening in your nose is all of the olfactory or smelling neurons in your nose, like maybe a couple get contacted with a little bit to begin with, and then more and more and more neurons in your nose area are smelling this signal. So more, what am I doing, frequency? Yeah. So more and more that signal's coming at you. And so individual neurons are like, okay, I smell it, it sends out an action potential. And then it's like, the smell's not going away. I'm gonna send out another one. The smell's not going away. I'm gonna send out another one. So they keep sending on action potential after action potential, but again, of the same strength. So if that signal is just coming, 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 so can the action potentials be sent on and sent on and sent on. Okay, the second one is not only in your nose that smell keeps lingering around, but now all, like a couple of neurons in your nose are contacted then more and more and more and more. And so between the two examples, one is that each one, maybe a couple of them are sending out a signal and sending it, like maybe the ones like at the bottom of your nose, they're like, uh-oh, I smell something bad, I smell something dangerous, and they keep sending out action potentials. But then as you're breathing and as more of that smell's coming in, all of the neurons in your nose are smelling it. So everybody's sending on action potential after action potential after action potential. And so if the stimulus is strong, one, it's going to make a neuron do action potential after action potential after action potential, and then there is the ability of a lot of neurons to be doing the same things. Thinking about this in another context. So if somebody comes up to you and they, I mean, depending on how hard you're concentrating, but let's just say you're, in lab and you're not like super focused, but there's things going on and somebody taps you, just like this, are you gonna turn around and be like, I'm gonna kill you, right? If they tap you, usually we know that that means like, oh, yeah, what's up, right? So somebody taps you, it might activate one neuron in your arm. And so you're like, okay, my reaction is just like, okay, there's just one neuron, so it must be something, they just need my attention. But if somebody comes up to you and they're like pushes right hard into your arm, like really, really hard, then you might react stronger because both of these neurons are being activated, or maybe a whole bunch of them are being activated. Where neurons communicate is that area, what we call synapse, or the synaptic cleft. That little tiny area where there are receptors from another cell that are going to receive or not receive the signal from the original cell. So that's the synaptic cleft, that area in between. This is where the neurotransmitters are released or spill out of our original neuron. Nice term, neurotransmitters. And just like we've said for many of the processes within our body, that the compounds are going to move from high to low concentration via diffusion. And as long as that signal fits in with the receptor, it's going to stimulate the next cell to do something.
Some of the book it says bind to postsynaptic neuron instead of bind to receptor.
Oh, well, yeah, that works. I think I didn't change it in here. You have the postsynaptic means the end of the synapse. So this is what we're talking about, is that if you're looking at all the way at the end, so we're looking at our neuron here, and it's receiving information from another neuron, that this is the synapse of this neuron, and then these branches are the dendrites of this current neuron. So we're going to take a look at this area up close, that from where the signal is being received, these neurotransmitters are shaped like a circle, and as long as that circle can bind with the receptors of this new neuron, then it can send, then it stimulates the postsynaptic, meaning after synapse neuron. So we would call this the presynaptic neuron, and this is the postsynaptic neuron. Pre means before, post means after. All right, so a little bit about some neurotransmitters. We hear a lot about neurotransmitters, especially nowadays. I like that we are having more focus on mental health issues. We're hearing about dopamine. Oh, so much about dopamine, right? The dopamine rush from being on your phone. The dopamine rush from buying something new. Serotonin, norepinephrine, dopamine, serotonin, norepinephrine, and what's the one at night that goes high? Melatonin. Those ones all cycle throughout the day. And so like dopamine, serotonin, and more norepinephrine signals go when you wake up, they start to ramp up, and then they're pretty high for a number of hours, and then they kind of dip down. some cultures, especially Latin American cultures have, and a lot of European cultures, will have a siesta somewhere midday, mid-afternoon. And the siesta really matches up with our neurotransmitter cycles, that they usually go through a cycle where after the morning, maybe mid-morning, they get pretty high and then they start to cycle down. Ever notice for yourself, there might be a point in somewhere in the early to late afternoon where you just feel like every day you feel kind of a little less energetic. That's when these are cycling down. And so a lot of cultures say, okay, everybody take a rest right then, because that's what our body is saying, is that we need a rest. And then they cycle back up, and then you have energy in the early evening, and then they cycle back down at around bedtime, and melatonin starts to go up, and melatonin helps you to go to sleep. So we do hear a lot more about these. There is so much research going on in these areas because our genetics individually are so different in terms of the cycle of many of these neurotransmitters, that some of us just have like maybe low dopamine, and low dopamine can have an effect on maybe being more depressed, more anxious. And so there's a lot of research, like how do we help people whose cycles are off? There's a lot of really interesting research about our phones and the way our phones are messing with these cycles.